Hello. So as you said, I'm Justin Porter. I'm in Greg Bowman's lab. As we heard this morning, it can be quite challenging to detect functionally relevant conformational changes in your large molecular dynamics trajectory sets. And one of the conformational changes that it probably won't surprise you after Greg's talk this morning to hear that we are interested in is these cryptic pocket openings. So this is beta lactamase we saw this morning. It's the active site serine. And there's this, is that mic on? I guess every other speaker has asked that too. It sounds up here like it's not on. And then there's this cryptic allosteric site here where there's a drug that binds, displacing this helix, opening this and modulating activity. And what I want to talk to you about today is a method that Greg and I have developed to try to discover this kind of behavior in a large molecular dynamics trajectory set, and then how we use that method to detect what we think are some other cryptic allosteric sites in the same protein, as well as look around for other sorts of motions that might be happening at the protein surface. And so this is the sort of classic molecular dynamics, or Markov state model sort of picture that we always see. And so the question is, if this is our big old Markov state model that we built sort of in an unbiased way, or sort of like a geometrical clustering, how can we pick out the yellow and blue states to see what interesting behavior our molecule might be engaged in? Unfortunately, Markov state models actually look more like this once you built them, and it's probably more than even the 13 dimensions that Benoit can imagine. Certainly, it's too much for me. And so you can pipe that into, for example, something like a pocket detection algorithm. And that works, and we've done that. But you wind up with somewhere between 5 and 15 pockets per structure in one of these states. And so now you have maybe 5,000 states and 15 times 15 pockets per state. And there's still a lot of data to sift through. And so what Greg and I thought is, can we sort of ask a different question that makes it a little bit easier to sift through all the results that we get? And what we're going to do to do that is we're just going to look at the solvent accessibility of residue side chains. So I think we probably all know what that is. It's pretty self-explanatory. Every atom has like a bit of a surface. When you roll a ball over it, it sort of owns that surface, and that's its solvent accessibility. And the important feature that we're going to look for here is correlations between residue side chains solvent exposures. And the reason that's useful is because pockets, they don't kind of randomly open, they sort of pop open. And so you're sort of seeing that here, is that all these residues in that deep pocket that we saw before, they open simultaneously, right? So they all change their solvent exposure together. In particular, you can sift through either all the frames or the states in your Markov state model and ask, what is the mutual information? It's a nonlinear measure of correlation. What is the mutual information between every pair of residues side chains? And turn that into a sparse graph. And so it kind of looks like this for this protein. And so your eye can probably pick out some communities here. Also, the colors help. The colors come from a graph clustering algorithm that we applied. And we call those exposons to separate them from like every other kind of clustering that we wind up talking about. And so you can assign, with this graph clustering algorithm, the exposons as labels to each of the nodes in this graph. And turn around and map that back onto the apocrystal structure that we saw at the very beginning. And so you can see that as like a nice sort of sanity check. We have discovered, in fact, that near things are near one another still. So there's the active site here that we plot in blue. We also find this known allosteric pocket here in light blue, as well as a pocket here that's been previously reported. And so that's nice that we are able to sort of re-find the things that we knew were there. But if we're looking for new pockets, we also want to know what the open state looks like. So it's not just sufficient for our purposes to assign residues to groups, but rather also we want to know which states or frames are open or are doing the thing that we're looking for. So the way we've sort of settled on doing that is we'll take our Markov state model and we'll compute the slow motions. This is the eigenvectors and eigenvalues. And look for which eigenvector best correlates, so which eigenvector's weights best correlate with changes in solvent accessibility of either the entire exposon or a particular residue within the exposon. And then extract the extreme structures from that motion, and then just make a morph out of them. a morph. There we go. And so you can see that in this case, with a known allosteric site in light blue, we've found something that looks awfully like the motion that we saw when we in the ligated crystal structure. And so that's nice. It would help us understand what an exposon is doing. But we'd like to be able to compare this or what we've discovered with an experiment if we don't already have a ligand to get a co-crystal structure with. So the way we've been doing that in the lab, and Greg alluded to this, is we have a thiol labeling assay. It's a biochemical assay where we'll mutate one of these residues to cysteine, spike in vitro a labeling reagent that has a sort of a colorimetric readout, and watch as that reaction happens over time, and compare that to either the sort of near instantaneous rate that we see, the unmeasurably fast rate that we see when a residue is exposed to the solvent all the time. And then the very slow rate that we'll see that's sort of consistent with the global unfolding process of the protein if it's buried. And so I'm showing green is surface exposed, red is completely buried, and then yellow is sort of intermediate rates that are an order of magnitude or more faster than what would be consistent with the global unfolding process, but also much slower than we would expect from something that's just exposed to solvent all the time. And so we can ask, do we find, for particular residues, do we find them to be exposed or buried in our Markov state model? Of course, if they're always exposed, that would be like a surface residue, right, in both the Markov state model and in real life. And then if a residue is buried, we never see it exposed. Presumably it exposes in real life as the part of the global unfolding process. And then compare those two things between our Markov state model, our simulations, and our in vitro experiments. And so you can see from this table that we get the surface and the buried ones correct and miss one exposure event from residue 249. It's down here. Interestingly, that's the residue that we think is reporting on the orange exposon that I showed you from before. So we seem to be able to sample some opening of that pocket, but simply missed, presumably due to sampling, its full sort of opening behavior. You can also ask, are we able to sort of get the rank order of the rates here correct? Because they differ substantially. And the way we sort of answer that question, module all the things that we just talked about with discretization and rates, you can take your transition probability matrix, zero out states that you think, turn them into sync states where you think the residue is exposed, prepare a system in equilibrium, and just keep multiplying by the transition probability matrix and see sort of decays as all the probability density winds up getting stuck in sync states. And you get sort of figures that look a little bit like this. So no labeling would be something where the probability stays always in states that are buried. Instantaneous labeling would be sort of analogous to a situation where the residue is always exposed, residue is actually irritated. And then you have sort of these intermediate pocket labeling residues. And this is, of course, an in silico result, but the table that I showed you before was ordered from fastest to slowest, and so I can say that the rank order of these guys is correct. Right. So that's like our retrospective analysis there is looking reassuring, but we'd also like to be able to prospectively identify things, right? That's sort of the point here. So we thought we could just go back to our exposon map here and pick out a new residue, or a new exposon that we hadn't thought about before, and characterize it in the same way that we characterized the previous ones. Specifically, we chose this beige group here and did the same thing. So we look at the exposon here. We see there's supposed to be a nice opening motion here. I don't know what happened to that. Hold on. Maybe it'll go if I do that. Nope. So we mutate this pink guy here that's hard to see in this part of the morph to cysteine, spiking the label, and find that it in fact does label as a pocket residue. This is a little bit of data that you saw earlier in Greg's talk. Something that we're always sort of is on our mind when we do this is that we have to introduce a mutation. So we're in a very literal way not looking at the same protein anymore. So one way we thought we might be able to address that is to look around for a homolog of TEM1, which is the protein we've been looking at, that has a native cysteine that is buried in a crystal structure but participates in one of these exposons. And so CK79 fits the bill. It has a buried cysteine that you can't really see because it's buried. And didn't introduce a mutation, expressed it, and ran the same experiment. And you can see that, in fact, it does label in a way that's not consistent with the global unfoldment process. An interesting thing that you can do once you have labeled protein is ask, what is that protein like? So you can think about your labeling reagent potentially as something like a covalent drug, a little bit, that's forcing the pocket open sterically. And so if you take the labeled protein and then see, does this protein catalyze the degradation of beta-lactam antibiotics? In the case of CAM1, which I just showed you, there's actually, interestingly, a 4-fold increase in the catalytic efficiency of that protein in the labeled form, which sort of plays into what Greg mentioned earlier today about it may actually be easier to design activators in this setting than at least I thought a priori. And then in this protein, we actually see a 15-fold reduction in catalytic efficiency, making, at least by this assay, this pocket the best one we've studied. And so we're working now on trying to exploit that, maybe even to find a lead compound or something. As a brief tease in a completely different system, this is a little chunk of the protein myosin. It's a 700-ish residue motor protein. There's a drug here, OM, that's an allosteric activator, or at least thought to be. It's in phase 3 clinical trials right now. And you can see there's a pink exposon, or purple, I guess, exposon here that looks like it's not doing anything, right? It looks like nonsense. But if you go and kind of figure out, like, what does this exposon do, you actually see that the protein folds up and nestles around where the drug would be if it were in the simulations. So I thought that was very interesting. And sort of promising that it applies to a separate system. We were sort of coincidentally also, something that had been on our minds this whole time is when you are looking at solvent accessibility correlations, they don't actually all have to be sort of positive correlations that constitute collective exposures. So you can have collective closures as well as negative correlations. So if one residue is exposed, the other one's buried, they can flip. And so we were kind of interested in hunting around and seeing what other things might exposons be, what other motions might they represent. And so this is Ebola nuclear protein. It's important for capsid assembly and bioreplication. And so I ran the exposon pipeline on it almost on a whim and saw these, kind of like in the previous example, these distributed exposons, which, as I said before, like, we expect them to be condensed in space if they're going to be pockets. And so I actually thought that this was a bug of some kind. But I went to go extract the motions that these exposons are reporting on, and we actually found something like this. This motion, it turns out, is consistent with hydrogen-deuterium exchange data that you can't explain using the cryo-EM structure alone, and is thought to potentially be important for viral replication and the way in which this protein is regulated by the presence of RNA. And so we thought that that's a sort of an interesting example. It's not a cryptic pocket per se, but is an example of a functionally relevant surfacial rearrangement that we did not have to know about a priori, but were nonetheless able to extract from the simulation, really in like an afternoon once we'd done all the calculations. And so this is a sort of tantalizing example of what this approach towards analyzing protein surface chemistry might be capable of. So in conclusion, we think that exposons are able to identify sort of the sites of potential cryptic site formation, that exposons provide a way for us to extract the motion that is giving rise to these correlations, and finally that they might actually allow us to identify other classes of rearrangement that are happening at protein surfaces. So with that, I'd like to thank my lab and my thesis committee, our collaborators, Foley and home donors, the money, especially in our lab, Katie Mader, who did all the biochemistry that I talked about. We're unfortunately losing her to medical school next year, so that's making us all very sad. Thank you.
Yeah, so you seem to find pockets all over the protein. Mm-hmm. So I must have missed this, but how do you rate the sites? Like, which ones do you go after first to test?
So that's sort of an open question still. You can look at the strength of the correlations. You can look at, once you've identified like what the opening and closing is, like what is it? So there's...
Do you have a lifetime for each pocket or something?
Well, we get that rate for particular residues. So as the pocket opens, the residues don't seem to share rates because there are many, many structures, each of which contributing more or less to sort of the labeling behavior that we observe. So right now, we're sort of bringing our biological knowledge to bear when we pick a pocket to go characterize. In the case of CTXM, for example, we wanted to try the cysteine one. In this case, we picked one that was near the omega loop, which we think might be important for catalysis.
So you just use your biological intuition.
That's what we've been doing, although I'm certainly interested in if people have ideas. You can look at the strength of the correlations of the total information that you find in a pocket. So each residue has associated with it, the amount of correlation that it has sort of globally. But it's not obvious to me that should or shouldn't tell you how relevant the species that it's engaged in necessarily is. Other questions? In the back. Oh, John.
So are the rates you get for this labeling proportional to the free energy in some way? Or I know you simulated it to go the other way, but can you do the inverse of that and ascribe some population or a free energy cost for revealing that allosteric site?
So you could certainly look at all of the states where you see the side chain that you're interested in exposed and just sum up the equilibrium probabilities for those states would be one way to estimate that. I'm not sure what your reference state would be in that.
The actual labeling experiment. Comparable free energy.
So
Or is
let's
the
see.
ordering just export it?
We use some of the machinery that was developed for HDX to analyze those data. Sometimes we have enough resolution to fit all three, open, closed, and labeling rate, but it turns out to be challenging for a variety of reasons. So I don't know of a case where we specifically have a cost that we've been able to estimate experimentally, although that would be very interesting. I definitely agree. Yes?
So also a quick one about the bioassay. So when people try to model binding of covalent drugs, they would normally look at how varied the cysteine is, but also the nucleophilicity of the cysteine for PK. So have you thought about this and how does this affect?
We have thought about that. I'm not a chemist, and so my thoughts have been extremely rudimentary. And we kind of were able to get the rank order in this case, and that was sort of good enough for us. If you have ideas as to how it might incorporate, so one example would be that we model labeling as perfect, completed instantaneous as soon as you're exposed, right? Whereas obviously the reaction takes at least some amount of time to occur. It's probably dependent on the complex geometry of the exposed cysteine. There are probably other factors I'm not thinking of, solvent, that also matter. I don't know how to model that, but I think it would clearly take the work to the next level if you were able to do that.
Have you thought of using this method to find cut sites for protein degradation where the industrial application is to stabilize the protein?
That is a great, I had not. That is a great idea. I don't know, do you know how, is that comparable to an unfolding event when that happens? Or
often one presumes that these cut sites have to be exposed for the protease activity, but often that's inferred.
Right.
And if you could actually find them, but prospectively figure out how to mutate the protein to stabilize,
that's interesting.
That's the industry application.
So we definitely are thinking about some of these even more excited states than just pockets in the lab. In fact, it's not submitted yet, right? Greg's your grant on the No. Okay. Well, I'm working on a grant right now, studying some of this proteolysis stuff, and actually the angle is how that might affect. I hope I'm not tipping our hand too much here. How that might affect E. coli resistance and the sort of balance of this particular protein in the cell. And I think the way to address that would be to leverage some of Max's FAST code, which we'll hear about tomorrow, to really access some of these much more distant from the much more excited from the crystal state structure, or somewhat more than these. Because I would imagine that those unfolding events might be even less favorable than these sort of peeking out of the core, if that makes sense. But I do like that idea. I think that would be cool.
let's thank those
Thank you.
again.